mine site restoration at Curtin Uni in Perth, and I'm chairing this session. And one of the motivations for this session is, from my experience at least, is that restoration monitoring is poorly done or not done at all, and it's poorly done. The data is inconsistently collected, or it's lost, or the methods are not sort of keeping up with what's happening now. So, following Tane's example, how many people think restoration monitoring is done poorly here? Put your hand up. Who thinks it's done well? Okay, I was going to ask that person to leave, but that person doesn't exist. So, yeah, that's surprising. That's one of the few things people involved in restoration agree on. So, yeah, we all agree that restoration monitoring is done poorly. There's 5 talks in today's session. 3 of them are around metabarcoding, and I'll introduce that as the first talk. And then we've got a couple of talks on drone-based monitoring. So we've just had a paper accepted on this topic, DNA metabarcoding, a new approach to fauna monitoring and mine site restoration. It's just been accepted in restoration ecology, and it came out of an honours project from last year. And our motivation to write this paper was that fauna monitoring in particular seems to be done very poorly in mine site restoration at least. So it was work by Kristen Fernandes. Kristen couldn't be here today, so I'm presenting it for her. So mining, minerals and resources industry is the basis of many global economies. If you look at the brown shaded countries there, they're the countries where metal minerals are the largest export. But if you add petroleum into that, which has similar restoration issues to minerals and extractions, you can see that it's probably the basis of most global economies. And in 2016, a global analysis of the top 40 mining companies estimated the annual value of mining to be around $748 billion US. So it's obviously a big deal economically, but it's also a big deal from an environmental perspective. It has a large environmental footprint, and very recently the Resources 2030 Task Force document was released in Australia, and some numbers on the importance of mining to Australia: over 200,000 people directly employed, and over $200 billion in exports annually. So, as an example of the footprint of mining at a country level, this is from a recent trip to China. The photo, the footprint in China is 3.7 million hectares of land degraded by mining in China alone, which is around about the size of the Netherlands. So it's a huge issue in China. So the scale and cost of restoration means that we need to make sure we're getting more bang for our buck. Okay, we have to be really clever about how we spend our money, and to do that we need to monitor restoration. So why do we monitor? We monitor to identify whether restoration actions are working or need to be modified. We monitor to answer questions about new treatments, the return of organisms or processes. So, for example, we might try a restoration planting where we plant nectar-rich species and see if that impacts on pollinator communities. Do we get more pollinators, more diverse pollinators? So we can try different treatments and see what their effect is. And perhaps most importantly, at least from a mining perspective, we monitor to provide evidence to stakeholders and regulators that goals are being achieved. So that's really important from a mining perspective. And it's a bit of a hot topic at the moment, completion criteria. I think there's several talks perhaps going on now on completion criteria. Mining companies need to demonstrate to the regulators and other stakeholders that financial assurances and liabilities can be removed. But how do we determine when restoration efforts at a site are on the right trajectory and have reached a suitable threshold for the mining company, for example, to be able to sign off on that site, to relinquish that site? How do we determine that threshold has been reached? So some of the common goals set for mine sites following closure include restoration of structure, diversity and function of the disturbed ecosystem. And plants are the focus of this, and fauna are often neglected. And the restoration is really poorly understood. We assume that if we put the plants back in, the animals will come back in. We're not, completely sure about that. Okay, so there needs to be more of a focus on monitoring fauna in restoration. It's a bit noisy next door, isn't it? So why should we monitor fauna? Why should we care about fauna in restoration? So fauna play a critical role in they have a right to be there, but also fauna play a critical role in ecological processes, including pollination, nutrient cycling, soil aeration, plant composition, seed dispersal, and also pest control. So fauna are really important to get into your restoration. So we argued in our paper that we need a more holistic model of mine restoration monitoring, okay? We need to monitor everything from microbes to mammals, okay? And not just focus solely on plants. So just to explain the eDNA workflow, the metabarcoding workflow, and for those of you that are unfamiliar with metabarcoding, it's an approach that can rapidly assess biodiversity using genetic material from bulk samples or from environmental samples. So you can take it from a range of substrates. You can take it from scats, from soil, bulk arthropod collections, either in pitfall traps or vein traps. And you can also use water as a substrate for eDNA. Once you take those substrates, you extract DNA from them, and then you amplify target barcodes. Okay, so if you're interested in vertebrates, for example, on your mine site, you'll use a molecular magnet to pull out those vertebrate barcodes, which can then be used to identify species. Or you can use plant barcodes to pull out plant species, or you can use fungal barcodes to pull out fungal species. You can do all this on the one sample, okay? So if you've got a soil sample, and Mieke will talk a little bit about this later on, if you've got a soil sample, you can pull out plant barcodes from it, vertebrate barcodes, fungal barcodes, maybe vertebrate barcodes, you can exchange it, microbe barcodes, etc. from that soil sample. So perhaps we can use that soil sample as a source of biodiversity assessment for the whole site, if we can successfully pull out all those groups from it. So we amplify our barcodes of interest, and then we blast them against a database, okay? A sequence database. And that will tell us what organisms, or perhaps not down to species level, perhaps not down to genus level, depends on what sequences are in the barcode and the resolution of your barcodes, but it will tell you what's in your sample. So does that make sense, I hope? So this approach has been being used a lot in the marine space, and it's being used for biodiversity assessment, detection of invasive species, food webs in the marine space, but it's taking a little bit longer to catch on in the terrestrial space. And there's probably some good reasons for that, which we'll discuss later on. But in the marine space, it's increasingly recognised by regulators as well. So it's starting to take off. And just some of the terrestrial studies that have been conducted, and this is from Kristen's paper. It can be used for dietary analysis. It's been used to look at food webs. It's been used to look at trophic linkages, and it's been used to look at vertebrate communities, etc. different substrates used include feces, arthropods, bulk arthropod samples, so in your pitfall trap or your vein trap, soil, as I said, and water. So there's a range of substrates you can use. And as I said, it's been used to look at plants and inverts, invertebrates, etc. So there's a number of advantages with this approach to biodiversity assessment, but there's also a number of disadvantages, and we'll go through some of those. So the advantages are that data can be gathered quickly and can be readily analysed and verified by independent parties. And that's really significant in mine rehabilitation, as it adds accountability to restoration activities. It enables your data to be audited by a third party. Another advantage of the approach is minimises the challenges of morphological approaches in some faunal groups. For example, insects. In Western Australia, I don't know what the state of play is in other states, that there is maybe one or two experts in ants where we can go to for our ant IDs, but that's it. And we don't really have that expertise in other arthropod groups. Okay, if we want to use arthropods to monitor their recovery in a restoration site, we really don't have people to ID them. Another, so just an example of the approach is a study from China, I think it was, where they looked at thousands of arthropod accessions using a traditional approach, and on the right, and a metabarcoding approach on the left. And they got pretty much the same alpha and beta diversity estimates from both approaches, and the management outcomes were the same, but the metabarcoding approach took a little bit less than a third of the time of sifting through those samples and IDing each sample. So there can be significant cost and time savings with this approach. But I don't want you to go away from this talk thinking that metabarcoding is a silver bullet for all your monitoring issues, because it's not. We've got a long way to go with it. We're only at the very start of the process, working out what substrates to use, what information we can get out of those substrates, what assays we can develop, what molecular assays we can use, how effective they are, etc., etc. So we've got a long way to go. So some of the challenges presented by the method, DNA presence indicates that the local presence of the living organism. Okay, that can clearly not be true for a number of reasons. You can get vacant DNA, so you could get material, plant material, for example, blowing into your restoration site, or you could get a bird of prey flying overhead and dropping a dead animal onto your site. If you sample that, obviously it hasn't come from that location. And there's also issues with how long does the DNA persist in the soil. If we're extracting DNA from the soil and we're looking at plant material, for example, pulling out those plant barcodes, perhaps those plant barcodes have persisted in there for 10, 20, 30 years, okay? We don't know how long the DNA persists in the soil, so we don't know whether we're capturing diversity at that point in time. So another challenge with the approach is determining absolute population abundance from DNA metabarcoding, and that's improbable, and it's improbable for a range of reasons. All organisms shed DNA into their environment, but they shed DNA at different rates. Their DNA degrades at different rates. There's also different gene copy numbers. Some organism will have one copy of a particular gene, so you'll only get one barcode from it, but another organism might have 5 copies of that particular gene that you're using as a barcode. So there's lots of reasons why determining population abundance from DNA barcoding won't work. Okay, so we don't know if we've got 1 qual on our site, 4 or 8, okay? And it's unlikely we'll ever get to that stage because of those reasons I just mentioned. So another challenge is choosing the appropriate barcode and then developing robust assays to detect the target taxa. There's no one gene that will work across all organisms, okay? There's not one gene that will tell you that this is Banksia species A, this is mammal species A, and this is microbe species A. We don't have that, so we have to develop specific assays to pull out the information that we want, okay? And I think Mieke will talk about that a bit. And also in another talk, that Mieke is doing 2 talks for us. She'll be doing a talk for Kristen later on, and that issue, she'll discuss that issue. But we can get over those issues by designing PCR assays that are specific to the taxon that we're interested in. So we can overcome that issue, just requires a lot of effort. So perhaps the biggest challenge of all is how to ascertain ecological functionality. That's increasingly the goal of restoration. But how do we get that? By just detecting the presence of a species at a site. That's an interesting question to consider. So maybe using metabarcoding, we'll be able to detect markers indicative of insect pollinators or decomposers that have key roles in reference to ecosystems. But clearly we need to do a lot of work around that if we're going to be picking out a single organism or several as indicator taxa, okay? But it is a possibility for the future. So I think we all agreed that we need better tools to monitor ecological restoration, but metabarcoding needs further development and testing. And it probably should be combined with traditional taxonomic approaches in some situations, but we're not quite sure where the niche of metabarcoding fits in restoration monitoring at the moment, as we're just embarking on the journey of looking at it. So I'd like to hear your experiences with monitoring. If we can get as many people at this workshop tomorrow, it's at 3 o'clock in room 6, S201, which I assume is the building, perhaps, I'm not sure. Yeah, if we can get as many people there as possible from diverse groups, practitioners, regulators, researchers, that would be really good. What we want to do at the workshop is just get everyone together, split people off into 2 or 3 groups and try to identify what some of the key issues are in restoration monitoring and then how we should go about addressing those issues and then hopefully getting a manuscript out of it for somewhere like restoration ecology. So I have to acknowledge Kristen, who was the author of this paper, Mieke, Mike, Kingsley, Richard, Grant, and myself, of course. So, thank you. Say 1H? Yeah, okay. So already I'm the chair and I've already gone over time, which is not very good, is it? So next we've got Mieke van der Heyde. She's a PhD student at the Centre for Mine Site Restoration, and she is doing the bulk of the work and trying to develop metabarcoding, trying to apply it to restoration.
Where is it? Sorry. Where's my talk?
Just click.
Oh, there. Well, that didn't help. Okay, and 4.
So we've got 5 talks and we've got about another 10 minutes to at the end, so we can go over a little bit this one.
Okay. Slideshow. Okay, I hope you can all hear me. I am sort of losing my voice, so we'll see how this goes. My name is Mieke van der Heyde. As Paul said, I am doing my PhD at Curtin University with both the Trace and Environmental DNA Laboratory and the Center for Mine Site Restoration. So my first project in doing this PhD was to look at how do you apply metabarcoding to a terrestrial system if you want to look at holistic biodiversity. You have a lot of different substrates to choose from, and how do you evaluate those? So as Paul already mentioned, the process of metabarcoding is basically to take a sample, an environmental sample, extract the DNA, sequence the DNA of the barcode that you're interested in, and then from those sequences you can determine the biodiversity. Now, there are limitations, but it's already proving to be a very valuable tool in biological auditing for a lot of different studies. There's a lot more research being done in aquatic systems because, for example, water is everywhere in aquatic systems. You can easily sample the water. And the DNA in the water is somewhat representative of the biodiversity there. But you also have other substrates in aquatic systems, like the sediment at the bottom, which can have very different communities from the water. The same sort of thing happens in terrestrial systems. You have substrates like soil. Now, traditionally, soil and metabarcoding have been used to look at soil microbial communities through fungi and bacteria, and that's been done for at least a decade. It has also been used successfully on occasion with things like vertebrates, plants, and invertebrates, but there's a bit of a geographic bias in these things because they tend to be in Europe and North America or New Zealand in temperate climates where the soil is very different from what we find here in Australia, especially considering that UV, heat, and moisture will degrade DNA. So if you're thinking about the soils that we have in Western Australia, for example, where it's sand, and in the north it's rock and clay, does the DNA persist there at all? We don't know. Another substrate that people have been using is scat, which is very useful for diet studies, but you can also use it as sort of using the animal as a sampler of their local environment. So you can get emu scat, kangaroo scat, bird scat, all sorts of things, and it can detect both what the animal ate and the animal itself. And if you look at predators, you can often detect down the food chain the predator that ate the herbivore and whatever the herbivore ate as well. So that's an example for ghost bat scat, which is pretty successful. For bulk arthropods, you get the advantage of extracting from actual bulk specimens. So with environmental DNA, it can be degraded with UV and heat and moisture. But if you're extracting from bulk arthropods, you get a higher yield of DNA, higher concentration, and it's a little easier to deal with that, and you don't have to deal with low concentrations of DNA. As Paul said, the taxonomic expertise is decreasing, and you have a lot of undescribed species, a lot of cryptic species. So DNA may be a way to measure those in the meantime, if we haven't been able to describe them yet. One of the advantages of metabarcoding is you have a per sample cost rather than a per specimen cost. So the cost of a sample with 100 specimens is the same as the cost for a sample with 10 specimens. So if you try to get those morphologically identified, your 100 specimen sample would cost a lot more than your 10 specimen sample, whereas for metabarcoding, they're the same. Another substrate you have is just plant material. So if you collect a little bit of a leaf of every species in a plot and barcode that, you can get the DNA. And that can be useful because you don't rely on flowering time, so you don't necessarily have to go back several times. However, you don't really get abundance or percent cover. So in that way, the traditional plant surveys have an advantage. The other advantage of doing this with metabarcoding is that you can get non-experts to do some of the plant surveys, because they just have to pick a leaf, they don't have to identify anything. So the aims of my study were to evaluate these different substrates, see where they overlap and what they can detect. So I had 5 different substrates because I divided the bulk arthropods into 2 traps. Vein traps, which trap the flying insects, and pitfall traps, which trap the ground-dwelling insects. Then I also had soil, scat, and plant material. So I looked at 2 different sites, one in the Pilbara, which is in the northern part of Western Australia, and it gets extremely hot. You have a red clay and rock soil, and it's typically spinifex grasslands. The other side was in the Swan Coastal Plain, which is around Perth. You get sandy soils and Banksy woodlands. I collected those 5 substrates. The pitfall trap was 4 different traps, made 1 sample. The vein trap was 1 trap, made 1 sample. Soil samples included 5 different subsamples because the soil is so heterogeneous. And then the plant material, a little bit of every plant in the plot. And scat was just walking around about 200 meters in the sample area to look for scats. And there were 20 samples per site, so it's a total of 200 samples. So I extracted the DNA, I amplified certain barcoding regions, and for this section, I was looking at a whole bunch of different barcodes, and these ones on the side are the ones that I found that worked. pretty well for most of my samples. So I've got two plant primers, two plant barcodes, the TRNL and the RBCL. I've got two invertebrate barcodes, MZART and N236, and I've got a vertebrate barcode. You often want to use multiple barcodes because there are biases in the barcodes. For example, the MZART primer set doesn't really pick up Hymenoptera, so we had to develop the N236 primer set in order to detect our Hymenoptera, which we knew was there because we saw so many ants. So once we did that, we sequenced the barcodes that worked, we did the bioinformatics, and we determined the taxonomy using reference databases. So plant material was detected in pretty much all the sites, all the substrates, at both sites. And I got a lot of OTUs from this. OTUs are operational taxonomic units. It's where you get the sequences and you cluster them into 97% similarity. And you can have multiple OTUs in a species, depending on how variable that species is. In total, we got about 260 families, if you put them all together. Vertebrate DNA, I only really found in the scat samples. I couldn't find much in the soil samples. Most of it was just undetected in my PCR, which was kind of disappointing. But it does speak to maybe it's just being degraded by the heat. The invertebrate primers I used on the invertebrates, the vein traps and the pitfall traps. So with the phylogeny, we got a lot of plant DNA, we got a lot of invertebrates that were ID'd, and we got some vertebrates. Some of the OTUs weren't identified, and that's because if they're not in the reference database, you can't determine what they are. And that's a problem, especially for Australia, because you have a lot of unidentified things and things that aren't uploaded into the database. It's a bit of a geographic bias because a lot more people are working on this in Europe and North America. So they have a lot more of their biodiversity in reference databases. So for our plant sequences, it's actually quite interesting because if you look at the plant and scat, they both have the highest richness, OTU richness. And then soil is the worst, unfortunately. And then the vein trap and the pitfall trap are somewhere in between. For the clustering, they clustered by their site and substrate pretty clearly, except for the plant material from the invertebrate samples tended to be more spread out than the plant material from the other samples, which clustered really well depending on their site and their sample. For the vertebrate sequences, you didn't get the same. The Swan Coastal Plain tended to be higher in richness than the Pilbara because Swan Coastal Plain is a bit more biodiverse than the Pilbara, but with the vertebrate sequences, they're pretty much even, and this is because of the bias in what scat you can see to collect, right? Generally, it's a lot of kangaroo scat because that's everywhere. You also get emu scat in the Swan Coastal Plain, and you get other bird scat that's easy to see in the Pilbara because the soil is red and bird poop is white. So you can get quite a lot of the diversity from that. You can see some of the invasive species, like the camels, the Camelus dromedaris, and the foxes. But you also have to be careful because you can see in this tree there's the Ardiotis cori, which is an African species. So it's far more likely that this is the Ardiotis australis, which is the Australian bustard, rather than the cori bustard. So it's a word of caution if you're using metabarcoding. You have to look at your species pretty carefully. Because if they're very similar and you don't have the right species in the reference database, it will match to something that's very similar. So you can control that with your filtering and making sure that you're looking at the whole barcode and not just part of the barcode and the similarities. So with the invertebrate sequences, you again had that pattern of more richness in the Swan Coastal Plain than in the Pilbara. And again, they clustered really well depending on substrate and site. And they clustered separately even though they're both at the same site. So they're obviously collecting very different insects. The Venn diagram sort of shows where they overlap and where they're quite different. Both the vein and the pitfall traps had about half of their families that are unique to them, but they shared some of the others with the other samples. Soil was a little bit disappointing because I only got 12 families from that, but there wasn't a lot of plant DNA in the soil. And this could be for a variety of reasons. I'll get into that in a second. Plants, you got a lot of different families. Again, about half of them were only in the plant material. And scat is very similar. So for soil, detected very few eukaryotes, no plants and vertebrates. And this is possibly because the UV and the heat degraded everything. And it's much stronger in Western Australia than it is in a lot of the places these other studies looked at. There's also more background, and I collected in early summer, and in Pilbara, the temperatures were really reaching up to 40 degrees in the day. And I collected from the surface of the soil because, I mean, if you look at the soil in the Pilbara, it's rock. It's hard to get down there. And there are some possible improvements if you want to use soil to look at the whole DNA. You could look deeper. You could pick a different season and extract from larger quantities of soil. That could help as well. So for SCAT, you had a high per sample richness. Overall, less families detected. So it seems like it's pretty good for looking at general areas, but of course they're not going to eat everything in a plot. They're going to pick what they want to eat. So they'll always be missing some families. But it may help to indicate some connectivity in the landscape through diet. For bulk arthropods, I had vein traps and pitfall traps, and they collected very different things, so they can be quite complementary to if you want to look at more broad invertebrate biodiversity. There was way more specimens in the pitfall samples, but that's also because you had 4 pitfall subsamples for each pitfall sample, whereas for the vein trap, I only had 1 vein trap because they're kind of large and bulky and hard to transport. So for plant material, half the plant families only showed up in the plant material sample. Other samples can't really completely sample the plant community, but you have the other thing of plant surveys can be quite efficient themselves, and they get the abundance and the percent cover data that you can't get from metabarcoding. So in conclusion, the more substrates you use, the more biodiversity you get. It's quite clear on that. The appropriate substrate will depend on your target organisms or the goal of your study. Soil needs a bit more work if you're going to use it, a bit more validation. Pitfall and the vein traps are quite complementary, and the SCAT and the plant material will also get a lot of plant material. So I'd like to thank everybody. Thank you.
Thanks very much, Mieke. We've got time for one question.
Yes.
Thanks for the lovely presentation. I think it's very exciting that potentially we have these tools. And the soil one to me is perhaps a non-starter because we're taking such a small sample. I'm assuming it's representative, it's not like a marine sample, another plant one. But we use the pitfall traps and the vein traps. To me, using it all in farming, and then what comes out of that, macerating it up and running a test, would be wonderful to have. I know people have done it, but that seems like logical way to do it. You can pre-sample with all the analysis in soil. So I didn't feel physically as you were in soil.
Well, if the soil worked really well, then it'd be really nice because the problem with the traps is you have to go twice, right? You have to go put them out, and you have to go collect them. So it's a bit of an extra time in the field sort of thing. So if it's just soil, it'll work well. Yeah. That's another option.
But the issues we have is that we collect those with these samples. And as you said at the beginning, nobody can identify them.
Yeah.
So you end up throwing those of them away.
Yeah, rather than throw them away, why not use them? Yes?
What's the cost?
It depends on which labs you go to. So some labs are like $50 per sample, and some labs are a bit more. It depends on a variety of things. For example, in Trend, they do a lot of trace DNA work, so like in archaeology. So we tend to be overly careful with a lot of things. We only use tags to identify our samples. We only use them once, and that can be kind of costly. So it depends on which lab you go to and how careful you want to be.
The costs are coming down. Sequencing costs have just dropped dramatically.
They used to be several $100 in some samples. Some samples are just $10 per sample.
Great, thanks, Mieke. Next we've got Todd Buters, who's another CMSR student, and he'll be talking about the use of drones to monitor seedling emergence.
Jesus Christ. Do we have a clicker and pointer at all?
I think this room's probably the space bar room, sadly.
Okay, afternoon everyone. As Paul said, my name is Todd Buters. I'm looking at monitoring seedling emergence and early growth with drones. Now, the reason why I'm using drones is pretty simple. They're, well, I wouldn't necessarily say cheap, but compared to some other methods, they're quite cheap to buy and they're very easy to operate. You can get very high resolution imagery from them. They can carry a variety of different sensors. Most of my work has been focusing on just regular colour cameras, but they can also carry sensors like multispectral cameras, thermal cameras, Lidar, potentially hyperspectral as well if they're large enough. They have extremely low turnaround times, especially compared to manned aircraft and satellites. They can cover a lot more ground than foot surveys, and they're actually extremely flexible as well in their deployment. There's no infrastructure needed to launch or land, and once you've done a flight, you can just land wherever you took off from, swap a battery out and head up again. And they can also be used to create very high resolution orthomosaics. Now, what an orthomosaic is a single large image that's been stitched together from multiple photos. The scale is uniform within the larger photo, which is very useful because it means any 2 points, if they look like they're the same distance apart on the photo, then in reality they will be the same difference apart. They can be made from GPS tagged images, so you can determine the exact locations of any points of interest within the orthomosaic, and they can provide a very wide range of understanding of an area. So by looking at a bigger picture of the whole area rather than a series of photos that only show a small amount, you can gain a greater understanding of what's actually going on. And because they're stitched together out of such high resolution imagery, you can still zoom in and be able to see very small details. They can also be constructed with a digital elevation model, which essentially, it's exactly what it sounds like. It shows the elevation of each point within the orthomosaic. Now, this is just an example of my first trial area, which is, as you can see, 4 different areas that have been seeded with various plants. You can see the entire area, which is about a roughly 25 by 25 metre area. And you can still zoom in and get a very high level of detail, even though you're showing such a wide area. Now, what I was looking at doing was identifying the specific species of interest within a certain area. For this trial, this was just lupins because they're cheap, they're quick to grow, and they're quite morphologically distinct from the grasses that we also sowed in the area. Now, the goal was to identify them and track them as they grew. Initially, it was extremely easy because the lupins actually grew before the grasses did, so it was a very simple process of if it's green, it's a lupin. When grasses started coming up, it became a lot more difficult. You couldn't just say, if it's green, it's a lupin. You had to figure out something else to say exactly how you were going to discriminate between them. So we needed something better than just looking at whether or not it was green. And so we looked to use object-based image analysis for it. Now, image analysis has generally been done on a pixel-based regime. Essentially, you look at the colour values for each individual pixel and classify them based off those colour values. Now, while that is useful, it doesn't really give you the whole story of what's going on because, as just from this project, green doesn't necessarily mean you can determine what it is. It could be any type of green plant. It could even be a piece of green plastic that someone's left lying around. So when we're looking at object-based image analysis, the image is split up into multiple small objects, and these objects can then be classified based not only off the colour, but also off the shape, the size, relations to nearby objects, and it's just generally a better way of classifying images than pixel-based. Another issue with pixel-based is that because it's based purely off colour, you can sometimes get what's described as a salt and pepper effect, where you have, just as an example, let's say you've done a flight over a suburban area, you're trying to classify what's rooftops and what's vegetation. And on one roof, you have a single leaf that's fallen on the roof, it's been picked up, it's been seen as green, and so that's classified as a tree. And you obviously don't have a tree growing through a roof. So with object-based analysis, you can take the context into account. And what I mean by context, I'll just explain over the next few slides. This is just a screenshot from Google Earth, and it really doesn't tell you much at all. Does anyone want to take a guess as to what it is? It's water. But, you know, even if we picked that up on pixel-based analysis, even if that was picked up as the right colour to say, yes, this is water, what is it? We have no idea. As you go out, you can see you've got some greenery to each side, so it's obviously not an ocean. But we still don't know, is it a lake or a river? And we zoom out further and we say, yep, it's completely enclosed by land, it's a lake. And so that's what I mean by taking context into account. If you were doing this with object-based image analysis, you would just say, okay, this has been classified as water based off the colour, but based off the fact that it's completely surrounded by land, we can put this in the class of lake. So you create rule sets like that to help you figure out what exactly you're looking at. And this is just a very basic examination of the sort of rule set that I created to segment out the lupins. Now, the initial step for any object-based image analysis is just to segment the image into smaller objects. That's done based off the weightings of the various layers that you have in the image, which for this one was just the standard red, green, blue, and digital elevation model. From there, anything that had the green ratio that hit the required threshold is considered a plant. And we merged all the plant objects that were touching each other together, if they were spectrally similar, which generally they were, and then we resegmented. Now, the resegmentation was not to split it up into literally thousands of small objects like it initially did. It was just an attempt to segment larger clumps of lupins so that it would be classified as multiple lupins close together rather than one big one. It didn't work as well as I would have liked. I'm still working on that, but it has potential. Now, after that, we checked the area of the plant objects. If it was below the lowest threshold area, we removed it completely because that was just a single blade of grass, for example, and so we could say with confidence that's not a lupin. Anything above the higher threshold that we set, we could automatically say, yes, this is a lupin because it's the only thing growing in this area that would be big enough for that. We check the height as well, based off the DEN. If it's tall enough, we say, yes, it's a lupin. We rechecked the colour. Now, I'm just going to skim over HSI because it's something that I could probably spend about 20 minutes talking about just on its own. Essentially what it is, it's a way of representing the colour with single values rather than the ratios between red, green and blue that you normally have to work with. HSI was created, it's actually a really interesting story. It was created to express colour well so that a broadcast for a colour TV could still be interpreted by a black and white TV, and that's the I, intensity, that just shows the lightness or the darkness, and then the HS, the hue and saturation, determine the colour. Again, if it was below the lower threshold, we removed it. If it's above the upper threshold, assign it to lupins. Then another colour check of TGI, which is Triangular Greenness Index. Now, this is a vegetation index that was designed to be an estimation of chlorophyll content based solely off visible light. If anyone's ever heard of NDVI, Normalized Difference Vegetation Index, which is done with multispectral sensors, this is essentially a visible light only alternative to that. Again, if below the lower threshold, we remove it. If above the upper threshold, assign it to the lupins. And finally, we checked the shape. The grasses tended to grow out in all sorts of crazy directions, but they grew long and thin bits out in all directions, whereas the lupins were fairly compact and they expanded relatively uniformly. So if it was a crazy spiralling out in all directions sort of shape, we could say that's more than likely a grass. If it's compact enough, we said, yep, that's probably a lupin. And we got fairly good results. You should be able to see, hopefully it's probably big enough. You can see slightly different coloured areas, slightly different coloured bits of green. Some of them you can see a small shadow just next to it. It's generally been picked up pretty well. There was a fairly low number of false positives for most of the areas, although there are still some that have been picked up as lupins, even though they're not. A small one just about a third of the way across down the bottom there, as an example. And this is what it looks like a little bit more zoomed in. Now, this is one example of where the resegmentation has kind of worked, but not as well as we would have liked. You can see this sort of S-shaped bunch of lupins. Initially, that was all one large object, so if you want to count the exact number of lupins, you'd have to run a count on the objects, and that would have just come up as one lupin. It's improved slightly because now it comes up as 2, but obviously there's more than 2 plants in there. Now, in terms of accuracy, we got fairly good accuracy across the various substrates, ranging from 75% accuracy to 89%. Generally, false positives were kept fairly low, except for tailings. One thing to note, though, is that the tailings, in terms of the actual values of the substrate, in terms of the red, green, blue values, were very different to everything else. And this is based off a single rule set that's been designed to cover the whole area. If we were to rerun this with a specific rule set for each substrate, I have no doubt that we'd be able to cut down on the number of false positives. Now, this is something that wasn't really the goal of the project, but I thought was just really cool. You might be able to see in the top image lots of little white dots, and they're picked out in red on the bottom. That's actually the lupin seeds. We were able to pick those up from an altitude of 15 metres with the drone that we were using. So I just thought that was really interesting and wanted to share it with you guys, even though it wasn't the goal of the study. Now, excuse me, not used to talking this much. Just a little word on resolution. In terms of what we were doing, we were flying between 5 and 20 metres height because we wanted to just, first of all, see if it was possible. The resolution that we were getting in terms of the ground sampling distance was between 0.16 centimetres per pixel and 0.63 centimetres per pixel. The resolution that you get is affected by a lot of things like the sensor width, focal distance, height above ground. What we need to do, and what will be done in future, I imagine, is get higher resolution cameras so that we're able to get a clearer picture of what's actually going on. Just in the time that I've been using drones, they've gone from having dinky little 5 megapixel cameras on them up to 20 megapixel cameras for the drone that we were using. There are currently 100 megapixel cameras out there produced by a company called Hasselblad, which eventually we might be seeing those on drones. So going forward, we really need improved resolution to be able to get a more accurate idea of what's going on. And just a little explanation of why that is. If you have more pixels, it's simple. You can get a better idea of what exactly is going on. No one's going to be arguing that Mr. Mario on the left there is clearer than the one on the right. And it gives another example of it in this image here. If we imagine, for example, that this shape on the left is a cluster of lupins, not only are you able to get a clearer idea of its actual shape when you've got a higher number of pixels, for example, the second on the left compared to the far right is a much more accurate representation. But also with smaller pixels you can get a more accurate representation of the area as well, which is important because one interesting thing that we found is that at the 20 metre height, a lot of the grass just didn't show up at all. So it is entirely possible as well that there's a sweet spot where rather than going for high resolution, you just want a lower resolution that only picks up the things that you're interested in. But that will be a very niche sort of use, and in general, the better the resolution is, the better outcome you'll get. Now, I've put my email addresses up here because I'm sure that I glossed over a lot of stuff. This was very hard to condense down, but thank you for your time.
Thanks very much, Todd. One question that I have is, as drone-based monitoring, is that already taken up in WA, in the mining sector? Or?
It's extremely limited. Most people, there's nothing along the lines of identifying specific species. For most people, it will be, you put a multi-spectral camera on it and you say, this is the health of this vegetation. Or you put just an RGB camera on and say, you have 40% of your area covered by vegetation. So it's not at all specific.
Okay. We've got time for maybe one more question. Yeah.
How hard would it be to program seasonal changes in color, for instance? I've worked with clients who replicate grass coverage on rehab projects in summer or summer is dead. It's currently picking up very well. It's going to change the color of the wet season. So is that hard to if you have to do that and just say you're looping which is dried off in the dry season, but they're still there and color that? Is it hard to program?
It would be pretty easy, actually. You can have multiple classes within one classification. So you could just write a rule set for if they're healthy, if they're... one example that we have is mango trees. Apparently the leaves turn really red in one specific season. And so we've been asked to look at identifying those from a drone. So it would be fairly simple. We actually tried to kill off the lupins as well. And then within the week that we first started droughting them, we had a massive rainfall. So that didn't work as well as hoped. But yeah, it is definitely possible to do something like that.
An honour student from senior site, Kristen Fernandes. She couldn't be here today, so Mieke's kindly offered to have another go. And presentations on using metabarcoding to track restoration trajectories, and arthropods is the indicator.
Okay.
Thanks, Mieke.
Yeah, so this is basically an application of what I was talking about earlier to mine site restoration. So we have a restoration chronosequence, and as you probably all know, a chronosequence is basically just sites that are spatially distinct, but also to have different ages since the restoration was applied. And the goal of these restoration sites is really to get these 2 years, 6 years, 13 years, looking like the reference bushland at some point. So biomonitoring is crucial to mining restoration, or restoration in general, as we all know from David's talk and Paul's talk earlier. But there is definitely a focus towards plants because they're easier to measure, they don't move as much, they're easier to catch because they don't move as much, and easier to see. So if you're looking at arthropods, they can be really good indicators of change because they have short lifespans, so they change quickly with changes in the environment. They're very numerous and easy to sample. You don't have to get animal ethics approval because they don't have central nervous systems. And they often are focal taxons for ecological restoration. Like Jonathan Major did a lot of work looking at ants as indicators of ecological restoration. And in Australia, they're used a lot for that sort of thing. So if you want to look at the arthropods, metabarcoding is probably a great way to go with it. It can complement looking at the vegetation and everything else. It can be faster and cheaper and easier to scale once you've developed a working workflow, of course. So the key questions for this project in particular were, can we obtain holistic biodiversity from the invertebrate DNA? So we're getting the invertebrate, we're also getting the plant DNA. Does the plant DNA and the invertebrate DNA tell us anything about the restoration over time? Are there changes in arthropod diversity? Do they reflect what we see from the vegetation surveys at these chronosequences? And what are the indicator taxa across the restoration? Are there invertebrates that indicators of early restoration, late restoration, and the reference bushland? And what can that tell us about our restoration? So this was basically done at 2 different mines, one in Gingin, one in Yangara. They're about 50 meters apart, still in the Swan Coastal Plain area. So you have that Banksy woodland on very sandy white sand soils. And we have chronosequences at both sites, from 2 to 8 at the Gingin site and from 2 to 13 years at the Nyangara site. So basically we did, similar to what I was describing before, we had 3 plots at every site of pitfall traps. We took those pitfall traps, we extracted the DNA, we sequenced our invertebrate barcodes and the plant barcode, and we got our biodiversity from that. Also, because ants are particularly useful as indicators of restoration, we focused a little bit more on the ants, and we got them ID'd, and we created a sort of a custom reference database for those, just to make sure that our IDs were correct. So we got quite a lot of the biodiversity from that. We got mostly Hymenoptera, which is the bees, ants, and wasps, which you might expect. We got several spiders, flies, grasshoppers, springtails, cockroaches, butterflies, and beetles, as well as some of our plant sequences. Right, so if you look at ordinations like this, you can see from the vegetation surveys that they cluster more by site than by restoration age and reference. So the reference sites are circled in red here, and the youngest sites are circled in blue. So the vegetation surveys really showed us distinct difference between the Gingin and the Nyangara site, whereas if you look at the invertebrate DNA surveys, you see that the younger sites cluster together and the older sites are also close together. The younger sites also cluster very closely together. They're more similar, whereas in the reference communities, the plots tend to be further apart from each other. So as you get older and in your reference communities, you have more complex structure in your community, so each site might be a bit more different from the other, whereas in the restoration, they tend to be a bit more homogeneous. The species richness didn't really change. It was the composition of the invertebrates that was changing over time. We also looked at the plant sequences for this, but they had very little pattern at all. So what they're eating or what's being lost in the traps that we're barcoding didn't really reflect anything to do with the restoration. So maybe that's not as useful in our invertebrate samples to reflect the restoration. Also, since Teen was talking about this morning, we really want to make it as similar as possible to our reference communities. So we had our reference communities, and we calculated the Bray-Curtis community similarity to the reference. And we could see that with the age of the restoration, it was becoming more and more similar to our reference communities, which is a very good sign. We don't know if we've reached a plateau yet here. We probably need some older sites and maybe some more samples at different sites, because we only had 2 mines that we sampled in this one. But it's a very good sign that they are becoming more and more similar to the reference communities. So when we're looking at our indicator species analysis, we had a lot of springtails as an indicator of the young restoration, and springtails are also detritivores, so that would make sense. They're eating up the many things and doing some nutrient cycling, maybe helping plants establish. There are a lot of different springtails. There's an estimate of over, I think, 2,000 springtails. A lot of these are unidentified. So that could be an avenue for further research to look more in detail about springtails and their role in early restoration. We also had things like the Rhytodopenera metallica, which is a known indicator of disturbance, and this was found at pretty much all of our restoration sites. There was also Iridiomyrmex splendens. Sorry, I'm trying to remember how to say it. But that was an indicator of reference communities. So it might be, if you see those in your ecological restoration, it could be an indicator of success in your restoration, since they were very strong indicators of our reference communities in this study. You also had, interestingly, millipedes from an introduced order, Julida, that were indicators of our reference in our oldest sites. And this could be an indication that they're taking over a niche of a native species, or they're just like introduced and they're staying there, but they can't establish very well in the younger ecological restorations. So we're not sure about this one. Could be some further research into this one. So in general, we found it pretty successful. Our invertebrate communities were becoming more similar to our reference communities, which is what you want in ecological restoration, especially if that's your goal. In the future, we might want to have a few more sampling points over a wider geographic range with possibly different sampling types. And one of the limitations of our thing is that we looked at a specific region, specific gene region, because it has the most number of species in the reference database. It's not necessarily the best gene region to use to look at invertebrates. So if in the future people could update the reference databases with more and more reference sequences from other gene regions from these invertebrates, it would make using invertebrate DNA much more accessible to a lot of people. Also, using targeted assay development. So we had to create the primer set that we used for these because there aren't a lot that target the Hymenoptera very well. They're a bit recalcitrant when it comes to metabarcoding. Fortunately, the assay that we created worked very well, and hopefully people will be using it in the future as well. So, effective mine restoration monitoring should include DNA metabarcoding. Obviously, it should also include fauna surveys and fauna surveys, but I think DNA metabarcoding has a role to play and it can be very useful. Thank you.